Hello, everyone. Today, we're going to cover a fantastic paper showcasing what frontier neuroscience research is about these days. This paper from Sammons et al. Is a reaction, in some sense, to an earlier finding by Guzman et al. That hippocampal cells that encode memory, specifically the CA3 region, have very poor local connectivity to other cells in that region. In fact, Guzman et al. Found that around 1% of these cells are locally connected. And this contrasts the models, theoretical models of how these cells are wired, and also our previous understandings. So Simmons et al. Went on to challenge this by doing two things. First, they did a electrophysiology study of over 50 mice to try to map out these connections. Secondly, they managed to acquire a connectome of a mouse to also validate, based on structural information, how many connections does a particular neuron make to its neighboring neurons. And in their work, they were able to find that the connectivity rests somewhere around 8%. So 8% of the cells found in CA3 seem to be connected to other cells in CA3, not projecting to some other regions. And this is a very important finding. It shows not only that we can use these amazing connectome-projectome methods to advance our scientific knowledge, but also that the scientific knowledge itself can be updated, as some claims are peer-reviewed by other groups and together correct perhaps incorrect previous findings. So please enjoy, sit back, and I'll see you in there.
We've got a hippocampus diagram here. CA3 is one particular region of that. If you look at classical models of how the hippocampus stores memories, like they in this paper, they reference a bunch of papers by Edmund Rolls and Trevis and stuff. This is one of the ones I picked out here. CA3 is unique in that it has a ton of recurrent connections. It's got a whole bunch of connections to itself, and so it has been assumed for many, many decades to be an auto-associative memory. That it would be able to do pattern completion within itself. And in order for it to do that, it would have to have a lot of cells, excitatory cells, that make a lot of connections within the same layer. And people had thought that they had seen that, and they had developed models around that. But then other evidence has been coming in. And one of the papers that our paper today, this Sammons et al. Paper is referencing a lot, is this Guzman et al. Paper. It's reference 24 here. Let's see what year was that? 2016. And what they found, surprisingly, they did a bunch of multi-cell patch clamp electrophysiology, which this paper also does. And they found that the connections were actually not particularly, it wasn't that strongly connected. It was like 1% about probability of any CA3 cell connecting, any excitatory CA3 cell connecting with another CA3 cell. And that study was in rat. There's a bunch of arguments in this paper to say maybe that wasn't, but in any case, that was a science paper. It was a big a splash, and at least according to this paper, it went counter to a lot of the memory models that assumed that CA3 had a lot of recurrent connections, many more recurrent connections. And And that paper actually gave a model that tried to show how you could still get attractors and memory from such a underconnected CA3 network. Well, in any case, what this paper does is it uses two techniques. It uses electron microscopy reconstruction, and it uses the same technique that this other paper did, this Guzman paper of whole cell patch clamp, multi-cell, to recalculate the probability of a CA3 cell connecting with another CA3 cell. And by both of those methods, they get an order of magnitude 10 times more probability of connection than this Guzman paper did. This is in mouse. Maybe there's a difference there, but in any case, what I think I want you guys to take away from this is that these experimental papers, they can have tremendous ramifications for models of memory. The difference between 1% connectivity and 8% connectivity probability is enormous when it comes to how easy it would be to store multiple memories in the CA3. And in the hippocampus proper. So that's the key result of this paper. The key result of this paper is that there is about somewhere between 8 and 10% probability of one CA3 cell in mouse hippocampus forming an excitatory connection with another CA3 excitatory pyramidal cell. What I'm going to do is I'm going to go through the electron microscopy results. I'm going to go through the electrophysiology results that support this. And then they do a relatively simple modeling study to kind of put these results in context of how easy it would be to store thousands of memories in a hippocampus. Any questions so far? Anybody out there?
Yeah, what about this idea of the hippocampus as an index to cortex? Do you need some way of estimating that as well? And it was a difference in the Guzman versus the Salmon's paper?
The Salmon's paper only about CA3. Mm-hmm. It is assuming that there is even in their modeling work, they are assuming that there is a subset of cells within a memory that are going to be activated. They don't say where those activations are going to come from. They just activate them with a certain amount of current. So there are inputs from cortex. Like if you were to look at this diagram here, CA3 is going to get stuff from dentate gyrus. It's going to get stuff from entorhinal cortex. Those would presumably activate a attractor memory within CA3, but they don't talk about that. I don't know if the Guzman paper does, but I don't believe the Guzman paper does either.
Thanks.
Okay, so what they did, so going to the electron microscopy, I should have had the electron microscopy up. Actually, let me go ahead and look here. So they have the full electron microscopy volume, although not any of the tracing, as far as I can see, within that they use for this paper. So this is a WebNosos link to the actual data that they use in this. It's acquired on a multi-sem at 4x4 by the thickness of the slices were 35 to 40 nanometers. And it's a relatively big volume. It is about 1 millimeter by 1 millimeter of the CA3 region of the hippocampus, and but it's only 62 microns in depth. There is another paper that they have in, I believe it's just in bioRxiv now, that is looking at the CA1 region. We might want to consider doing that at some point. It's an even larger volume, but for this one, this is just in CA3. Okay, so so what they did is now they didn't trace the entire volume. That's not something they could do. What they did is they took this single volume. They found 7 cell bodies, seven pyramidal cells that, I will quote them, we identified 7 pyramidal neurons with at least 2 axon collaterals within the volume. So there's a certain selection bias here. I'm gonna put it out there. They don't really say anything more about that. This is a very thin slice, 62 microns. So they basically had to say, hey, we're not going to just take random cells. We're going to take 7 cells that are at least have an axon collateral that happens to be within the volume. Okay, 2 axon collaterals within the volume. Then they identify all of the outgoing synapses on those 7 axons. There were 1,062 of those. They followed the postsynaptic dendrites of each of those synapses out to either a cell body, 304 cell bodies, or until it hit the border of the data set. Okay, and then they said, hey, we only were really caring about excitatory to excitatory. So we are going to use the spininess, how many spines you have on these dendrites, to classify the dendrites, the postsynaptic dendrites that these 7 axons are connecting to, as either excitatory and inhibitory, and they found 795 excitatory cells. Okay. They had to also only consider the ones that had cell bodies within this volume, although I think that was the majority. It's a little unclear. And then what they did is they said, let's look at each one of these. So we've got cell number 3 of these axons. It contacts one of these other cell bodies. How far was that cell body? And they did that for each of these, and they've got these plots here. So the key one here I want you to look at is this found connections. This is saying the x-axis here is intrasomatic distance, so it's the distance between the 2 cell bodies, the one that had the axon that they're following, and the one that it made a synaptic contact with, right? And they plotted the number of found connections for each of these. And so within 50 microns, they found this number of connections, this number of cell bodies that this cell connected to. Within 50 to 100, they found this many. Within 100 to 150, they found this many. And they could compare that with the total number of cell bodies within that distance. And by doing that, they could divide these two and get the probability of connectivity within a certain distance. And the gray lines here are saying for each of the 7 axons. The black line is the average of all of them. And so if you look at the average of all of them, you get a I'll read it the way that they say it here. We found a local connectivity of 11.2% probability. That's a probability of any individual CA3 excitatory cell to synapse on any other individual CA3 cell. We found a local connectivity of probability of 11.2% within 50 microns of the presynaptic soma, the cell body that we're tracing. And that dropped to 5.5% within 250 microns. And the key thing is they said, to our surprise, this exceeds the previously reported connectivity by up to an order of magnitude. And this is this Guzman et al. Study that had been in the literature prominently for CA3 until then. So another thing that we should think about, though, is that this is a thin slice. And so these numbers should go up, if anything, because there's got to be some, as we go farther and farther away, we're going to get less and less connectivity simply because these axons go outside the volume that has been reconstructed.
Ken, can I ask a quick question?
Yes, please.
Yeah, I was wondering because I noticed that it was a pretty thin slice, and I was wondering if you have the information there on how it was oriented in CA3.
I'm going to let you follow up on that question because I think you okay. Probably have a better idea of what orientation means in CA3 than I do. So right back at you, Randall.
Thanks.
Did you recognize that they oriented it?
No, that's why I was asking. I didn't, but I have to be honest. I only read it very quickly, so I might have missed this somewhere. Yeah.
Just 400. No. Hold on. Electron microscopy. Thick horizontal slices with a vibratome. Does it say what the orientation was? Horizontal.
Horizontal slices. It would have been nice if they had just a low res image.
Like a map. Yeah, I agree.
Yeah.
They also in the electrophysiology that we're about to go to, these were also horizontal slices. So I kind of figured that these are probably so this is their schematic of it. So I would assume that this is the best axis for getting collaterals.
Yeah, that does make sense. Yeah.
Okay. All right.
Sorry, Ken, I have also a question. I'll put my head down. It directly ties into this. So the thickness of 60 microns. Did the authors, do you think, did this because there is an assumption that the neuroanatomy of these CA3 circuits is kind of restricted to some small layer, or is it just a physical engineering limitation? What do you think?
It's a physical engineering. In fact, the slices that they're about to use for the functional connectivity, those were 400 micron thick. And I've seen studies that kind of say, well, 400 micron thick, we're going to lose some at 400 micron thick. So this is purely a they had to slice 1,700 sections at ultra thin and make sure that they were collected properly. So it might very well be that they were trying to cut thicker and something screwed up in the slicing and they couldn't get it to work.
Okay, understood. Yeah. And then one small comment that I just wanted to get a clarification on. The outgoing local synapses, 1062, how many neurons do these 7 axonated neurons actually synapse to?
Yeah, so it is... Okay, so it's probably here because they say there's there's 1,062 synapses, and they say that there is 10%, so 96 out of 947. So it's 947. So 947 total connections, 96 of which are multiple synapses. So more than one synapse onto the same cell body, or the same cell, the same postsynaptic cell. There is one caveat with that, because if some of the cell bodies are probably outside of the volume, and so if you have a synapse on one dendritic tree and a synapse on a different dendritic tree, you can't necessarily know that those dendritic trees aren't part of the same cell if they go outside the volume. Does that make sense?
Thank you. Yes.
One of the other things I thought might be interesting, I just noted here, is they say they didn't observe any autopsies. They didn't observe any cells that connect to themselves. And actually, it seems to me like that is unlikely, given the statistics that they already found. It's like 11.2%. If you've picked 7 cells at random, they should probably at least one should be connecting with itself. Maybe not. It's not that big of a thing. Does anybody know if autopsies are a thing in CA3, or if they're avoided?
I do not remember, unfortunately.
Okay, I don't think that that's something that you could easily determine with electrophysiology. You would almost have to do that with this type of electron microscopy, if I'm understanding it correctly. Okay, so in any case, if this was the only part of this paper, I would still be super impressed, because it's a really big connectomic study and it's really cool. But I could see that reviewers would pick this apart and say, oh, you picked these 7 axons with a bias. So how do we know that this is really characteristic, right? This was only 62 microns. How do we know that it's characteristic? So it's really good that they did functional connectivity as well. So the functional connectivity that they did, they took, this is an overview of the experiment. They took a total of 52 mice. So instead of 1 mouse, they took 52 mice. This is a testament to how much easier electrophysiology is than connectomic studies. So they took 52 mice. They recorded 18 slices from total. These are 400 micron thick vibrotome slices. They recorded from 410 neurons with up to 8 cells simultaneously. And the typical type of experiment was that they would inject current into one of the cells, like let's say this blue cell here. They would inject current into this blue cell, so it's caused to fire, and then they would look for postsynaptic potentials in the other neurons that are potentially connected, right? And by doing that, they found these things. They identified 103 connections out of a possible 1,172 connections tested. Does that make sense? That's like, they're testing 8 at a time or up to 8 at a time, and they're essentially testing all possible. They're firing each of those cells and listening to the other cells, and they found 103 connections out of a possible 1,172. So that is 8.8% probability of any CA3 cell connecting to any other CA3 cell. They also measured some other stuff here. Let me go directly to this plot here. So this is in their supplementary. They were also able to test because they backfilled these with biocytin, I believe, and they could know where the cells were, and they could measure the distance between the cell bodies. And so they looked for whether there was a higher probability of connections to of cells that were close to each other versus far away from each other. In this case, they didn't find any. So if you look at the found connections, they got a certain amount in the 0 to 50 micron distance range, but there was a higher number of connections tested within that. And so when they go ahead and divide these through, they get essentially a flat curve, a little bit higher probability, but not significantly higher probability of two cells close to each other being more likely to be connected than far away. Okay, that's a little bit at odds with the EM, but not really. And the amount of connectivity is about the same. In this case, it's an average of 8.8%. Let's see, some things I wanted to say about this. They are able to measure the amplitude of the excitatory postsynaptic potential. But if you think about that, if you had a very, very weak synapse, like a silent synapse, you would not see it. So you can imagine that you would be able to see synapses in the electron microscopy that you would not be able to see by this technique, if I understand it correctly. Any questions on that? So essentially,
no immediate question. I'm just really stunned and in fact I was stunned reading all the way through the paper how much this is kind of like what you would love to find for, say, the predominant models of CA3. It's like,
okay.
Oh, fits perfectly. How is there nothing that's really off so far? Yeah.
Yeah.
I guess what
yeah.
okay, yeah.
Go ahead.
I mean, this is a question for later, I guess, but I hope that this group still has funding, all things considered.
Yeah, I mean, they're going into CA1 right now, like I said, in a bioarchive paper. So they're plowing ahead, and there's other groups. I've heard that the group at Princeton, Sebastian Sung, is got a CA3 connectomics project that's ongoing. That is looking at this with a TEM data set. Okay, so the other thing that they were looking at, now remember I wanted to ask Randall or anybody else that might know. So they really do reference this Guzman et al. Paper as kind of, gee, we found something different than what other people were expecting in the literature. Was there no, is that Guzman paper like an outlier lowball, and there's a whole bunch of ones that were also in the 10% range? Does anybody know?
All I know is that from previous electrophysiology, the assumptions were more along the line of the 8.8% that they found. And so the Guzman paper looked like a bit of an outlier from that point of view. And I was not under the impression that anyone really jumped on it and went, oh, the Guzman paper shows us everything is different.
Okay.
So this is kind of back to the default.
Okay, it's good to know, because I'm always, no matter how good the result is, I'm always kind of like, should we believe this? So the other thing that the Guzman et al. Paper had found was motifs, high order motifs, and they put a bunch of stuff because they didn't find the high connection probability. They had developed bunch of modeling based upon the higher order motifs that they had found. This paper looked for those higher order motifs, and my recollection is they basically did not find any significantly any significant higher order motifs. And I think that that's also like a regular. Isn't the idea that CA3 is supposed to be kind of simple? It's a fully connected.
Right. Yeah.
It's simple.
Yep.
Right?
Yep. It's totally
Okay.
in line with every standard model, basically. Yep.
Great. Okay. So that's the full experimental side of the paper. Then they do some computational modeling. And I'll go through this very quickly because I'm dying to hear what Randall thinks of the simplicity of this model, because I'm pretty sure that this is a way overly simplistic model of CA3. But what they did is they said, okay, we're going to assume that memories in CA3 are of this type. CA3 has let me go to the main figure of that. Okay, so we're going to assume that CA3 has N neurons. Okay, N is in Nancy, right? This big N here. We're going to assume that the memories are composed of random subsets of M neurons. So N might be 1,000,000 and M might be 1,000, right? We're going to assume that that memories are composed of M neurons that are themselves all connected as much as they can by stronger excitatory connections. Okay, and then because we know that the hippocampus is all about sequences, we're going to assume that there are that each one of these attractor states is also sending excitatory connections to the next M subset, and then the next M subset, and the next M subset. And so, if to test a memory recall would be to activate one of these, I'll just say memories. Activate one of the memories and then see if the sequence of memories replays. Okay, and they give an example. They have a computational model. It's a leaky, integrate, and fire model. Relatively simple, but not that simple. I mean, you definitely are having spiking neurons. You have N divided by 4. Inhibitory neurons. The inhibitory neurons are set up with a form of spike time dependent plasticity, so that it gives the proper amount of inhibition to put it into something that they call a what do they call it? Well, some kind of state that's an interesting state. So it's not overactive and it's not underactive. It has a way of adjusting the inhibitory automatically to get into a kind of an interesting state. And so they will form these memories, and I'll tell you in a second how they form these memories. They'll form these memories, and then they'll inject half of the cells within a particular memory. They'll inject those with enough current to cause them to fire. Presumably, they'll fire everything within that group, and then they'll go into a refractory period, but that'll cause the next set of neurons to fire, and the next set of, and the next set of. And so, if you look at plot B here, this is a sequence of those memories that are properly being recovered. And this is a sequence of memories that fails. Does that make sense? I didn't say that very well, but
So you were asking about the realism of the model, and
Yeah.
yeah. So it's not totally unrealistic. So the thing is, they're focusing completely on CA3, which means they don't have entorhinal input, dentate gyres, anything like that. They don't have any of that. They're not feeding it forward to CA3. They don't have theta rhythm. They haven't got any of that in there. So the only mode that they can do, the only, like something similar to what bigger, more complex, more sophisticated models of hippocampus will show, the only mode that this accords with is the ripple mode, where you get a ripple retrieval very fast, as you would, for example, in retrieving well-established memories, or say during slow wave sleep when you're kind of running through those as well. So you don't see a lot of the other behavior where you would say have kind of an acquisition phase and a retrieval phase, or where you would have a stronger separation between the patterns, the individual patterns that are being retrieved in sequence here. I also didn't see them testing cued recall, where you would give only a part of each pattern and then see if the whole thing comes back and how much percent the cue needs to be or anything like that. Yeah. But it's not crazy. You know, it's a basic ripple effect.
Okay. And I think from this perspective, it's also because one of the things I look at here is from a memory decoding point of view. This paper is showing how difficult it would be under this model to decode memories from the connectome, right? So the other thing is that I believe, and this is what they say in the paper, if this connectivity was not 8%, but it was 1%, this would be much more difficult to do. It would be much more difficult to find parameters. There would be a smaller space of parameters that could robustly store many memories than having a high connectivity. Where does the connectivity come in? Let me back up for just a second to that. So basically there's assumption of random connectivity of let's say 8%. They vary that, okay? But there's a random connectivity that they assume, and they assume that that's in a log normal distribution, like people usually see for synaptic weights, and they make the assumption that the strengthened synaptic weights will be chosen from essentially this 8% connectivity, and will strengthen up to the very top of the log normal distribution. So that's how they form these memories. They basically, which is also not necessarily the case if you think about it, right? Because you could imagine that there's 8% connectivity, but that 8% connectivity could be much higher within a memory, and they're not saying that. They're saying that they're making the assumption that you still have 8% connectivity even within one of these clusters, but it's just strengthened. I think that is a reasonable assumption, because
I don't think it's that crazy, because if you're trying to capture something that's coming through and you've never seen it before,
exactly,
you need to make use of existing connections. You basically have to have coincidentally co-activating cells, and then you apply some short-term potentiation and long-term potentiation to that. But you're not going to necessarily, build a whole bunch of new synapses in that moment. So it just has to be an existing set that you pick kind of at random, and that's where the 8.8% is really useful.
Exactly, and I think that's an argument that couldn't be made in, let's say, a sensory area that might have developed over, the full, early adulthood of the mouse or something. But in a hippocampus, it's supposed to be able to learn arbitrary patterns. So what they do is they have this spiking model, and they run it with a bunch of parameters. So if you look at C here, On the x-axis, they have different connectivity assumptions. So they go from 4% connectivity all the way up to 14% connectivity. And they have pattern size. This is assuming a network of 50,000 cells. I think the actual is 1 million for the hippocampus. They said somewhere else in the paper. But in any case, there's for different ranges of pattern sizes, they test whether they test the 6% successful replays. And anywhere in the yellow, it's unsuccessful. So basically, this percentage of connectivity and this pattern size does not allow robust retrieval of memory. But anything above this dashed line, anything in the blue here, is giving essentially 100% successful replays. And the 1 and 2 boxes here are the same network. 1 is showing a failure, 2 is showing a success. Right? So essentially, you can imagine if this connectivity was much farther down, there would be you would have to have much bigger patterns, and bigger patterns would be would mean that you could store fewer memories without overlaps, and it would just be less robust. So it's an important. They do some other things here that look at given a particular 8% connectivity assumption, what weight there, what weight of synapses works. So this is a sequence weight in picosiemens. So it's the connectivity from one of these memories to another, the average synaptic weight. So if it's not very strong, then it's not going to work. If it's very strong, it's going to work. If it's too strong, it doesn't work again. What they're essentially saying is, here's, under this assumption, there's a wide parameter space that that allows robust memory formation. So in summary, this is what it is. So they found connectivity of 8%. That's good. I think what we should also take from this is, if there was a connectomic study of the hippocampus, let's say with calcium imaging, where you wanted to prove that you had the ability from the connectome alone to find, to decode memories. This type of study would say how big of a volume, how many cells, how many synapses would have to be accurately traced in order to recover those memories under this type of a model. And that's why I chose this paper, because it ties directly into the memory decoding challenge. So that's all I got. The floor is open for discussion.
Hey, quick question, Ken, if you don't mind sharing again.
So I'd like to.
You are muted. Samuel is muted.
Sorry about that.
Oh, their hands? Is it hands thing? Oh, sorry, hands.
I just had a technical question about the last figures that you were showing. Would you mind opening those again?
Oh, sure.
As you made a comment about as the n of cells goes up, the memory that you can encode fewer memories and they would be less robust. And I just wanted
I
to
did say that.
understand that comment.
Yes.
So you were referring to figure C, I think.
I was referring to figure C. Actually, so figure C is definitely saying that you have to have this many, if you want to if you want to have robust retrieval, let's take a specific example. Let's say we have 8% connectivity. If we want to have robust retrieval, then the pattern size would have to be 1,200, let's say 1,100 or more.
Of M's.
So out of 50,000 neurons. So so along those lines, if you think about it, if you have 50,000 neurons and you're choosing 1,100 neurons at random, then how many of those memories can you choose before you start to get significant overlap between them? And so the thing that I wasn't making clear is that if you can avoid overlap with memories, then you have robust retrieval. But if you try to store more memories, so many memories that there's a significant overlap between memories, then you're going to reach the capacity. And so that's why I'm saying that the lower the pattern number relative to, the lower M is as a percentage of N, the more memory capacity of the network.
I think that makes sense. I will ruminate over this, maybe come back later.
Okay. There is a wide literature on this, and I know the Rolls papers that they were referencing, they have other things that come into this, like the number of synapses. I believe Rolls had, Edmund Rolls had determined that the number of memories was also capped by the number of synapses per neuron. So there's other things that come into memory capacity other than just connectivity, random connectivity percentage. Let's see, do they make a grand statement at the end here about that? Yeah, I'm sure they make a grand statement somewhere, but the other thing is that deep in the methods, they talk about a million N is equal. Here we go. The network size varied from in vitro like 1,000 to 10,000. So if this is a single slice, to in vivo like CA3 networks of approximately 1 million. So I think I'm getting from this that N in the real animal would be 1 million. Randall?
I think John was first, actually.
John?
Yeah, so I guess I'd love to know what is happening. What's the model for what's happening when you're replaying a memory, doing the pattern completion in their comp neuroscience model? Is that basically recalling a recent episodic memory that has been experienced? You're recalling it from a partial cue. What is actually happening in what they call replay? Because what I would like to do is I'd like to relate that, if possible, to this idea that information is somehow consolidated into cortex, right? I don't understand that part, and if they have any model for how their replay relates to that, because obviously you can't keep those memories there for long. So what is that thing that's happening in replay?
Yeah, I'm gonna let Randall tackle that because he's the expert on hippocampus here. So go for it, Randall.
I'm not 100% sure if I understood the question right, but there are two main aspects to replay in CA3. One is that you have to reactivate an individual pattern. So there need to be enough connections between the cells within a pattern so that if you only end up activating, say, 40%, 50% of that pattern, that very rapidly that whole pattern gets reactivated. And then the next stage is that you want to be able to activate next patterns in the sequence, because it's episodic memory, so it's sequence memory. So then you have to, that pattern needs to cue enough of the neurons in the next pattern so that you can then complete that entire pattern and then move on and on and on until you've gone through the entire sequence. But of course, that's just CA3. And in reality, you have other areas feeding into it. So some of that reactivation of a whole pattern within itself can already be happening, for instance, in entorhinal cortex layer 2 before you even enter CA3. So it's not entirely clear to me how much of this has to happen just in CA3 itself. CA3 is normally associated mainly with that sequential aspect, so going from one pattern to the next.
I see. The full replay of an episodic sequence.
Yeah.
Can I have a follow up on that before I get your question, Randall?
Sure.
And I think John might have been also asking this. So in some of the other systems that we've been talking about, like the lateral amygdala, there is a random subset of cells that are interconnected that are going to form the engram. And then there is an association that is made because of contextual fear or something. And so is it proper to think of this in the same way, that there is a random, not just a random engram, but there is a random engram sequence that is in the hippocampus but not associated yet?
Yeah, this was actually what my question was going to be about.
Okay, well,
yeah. So, and the answer is a little bit, the answer to your question is kind of twofold. So the answer is yes, believed to be some random set of neurons that form an engram of sorts. But it is also known that different parts of CA3 seem to be involved with different types of memories. So there are some that are more associated with, say, visual input, and others are more associated with, and it's not even clear, it's not as clear as that. So when you look at the work that Dong Song did, for example, in human hippocampus, testing which parts of hippocampus were activating for different types of memories that the patients were dealing with, it was something completely different. It was like memories related to people or visual cues versus objects. So different parts of that CA3 region. If you just follow it along, its architecture. If you go along the anatomical sort of length of the of CA3, you find that different areas are involved with slightly different memories. There seems to be some subdivision there, but then within those, it seems to be pretty much random. And my question was really associated with that, which is if you're thinking of a memory decoding challenge, looking at CA3, you'd have to have some kind of a protocol to establish if I'm looking in CA3 and I'm seeing this set of cells activate, that is associated with memory A, and then this other one's with memory B, and I can either distinguish them or identify them or something like that to suit the non-trivial memory identification or extraction criteria for the challenge, right? How would you go about that?
Well, this is a question for the group here, because one of the things that we're trying to get out of these journal clubs is a better definition of what would constitute winning a memory decoding challenge. So let me take the devil's advocate, which I don't think people will agree with, but I'm going to take it anyway here. Just for this, I don't agree with this either. But if you had calcium imaging that showed a sequence, let's say, of activity in CA3 in a slice, okay, and then you put that slice in glutaraldehyde, did the EM, picked out connected subgroups, and found that that sequence of activity could be predicted from the connectivity. Would that constitute a decoding of a memory? In
That's
that case,
what I'm wondering, right? Because then
Okay.
If you were to say you have to be able to extract or identify a non-trivial memory, then all you would have to do is find 2 or more ripples, in a sense. And you wouldn't have to know what they mean. They don't need to be associated with anything. You could do them in vitro like that and just find 2, 3, 4 different ripples that get reactivated.
But wouldn't you have to also connect it to some way the animal was trained, like in that lick right, lick left paper that you mentioned, Ken? Wouldn't you have to associate it with some external training that encoded a specific memory amongst the many possible ones?
Maybe. Let me again. Let me take the devil's advocate. This is not my opinion. Okay. If the goal is to address the fundamental concern that the connectivity or anything else that we can prove is preserved is getting the information that would be necessary to pull out memories and learn behavior, right? Then even if it's a subset that isn't connected, the implication is that if we did this and this other region and this other region at the same time, then we could have connected it to behavior, but we only did this, but we can extrapolate that from this experiment that we could have gotten it. And really it's saying, you know, are the memory models that we're basing our assumption on that connectivity is encoding memories, are those, if we test those directly enough, isn't that in the spirit of the decoding a memory challenge?
So is an alternative statement of the memory decoding challenge, is it then that you demonstrate that connectivity is related to memory function?
Yes, and I mean, so again, I'm taking the devil's advocate here, right? So, or I'm just, you know, there should be a robust debate that is happening within this community about what is really necessary, right? There should be a not just a I'm skeptical that the information is being preserved. There should be a I'm skeptical that the information is being preserved. Here is what I would like to see to convince me that the information is being preserved. And what I'm saying is that that's different from saying I want something that's sexy, that's kind of a memory that everybody, every lay person can kind of look at and say, yeah, that's a memory. Or a lay person could say, I don't know what CA3 is. I don't know what calcium imaging is. I don't know what these synapse things are. You know, if you're talking about a memory, I want to, you know, it's me blowing out a candle on my 5th birthday, you know, show that that's extracted. That ain't going to happen.
So Ken, the part that I like about it, obviously, is that, you know, it's a way of connecting, so the connectivity aspect, and then it's a way of connecting that directly with function and saying, in order to be able to have memory and identify memory, you can do that without, say, the quantum mechanical radio frequency communication between distant neurons or whatever. Yeah. So it looks at that. But when you're saying this means that the memory has been preserved, all you can say is we can show that some memory preservation is shown, that we have some evidence that there are patterns of memory that we can retrieve just by looking at connectivity, but you can't automatically say, and we haven't lost anything else. There are no aspects of memory or of those engrams that were not preserved in the connectivity. Which I'm not saying there is, but someone might critique it that way, right? They might say, okay, that's not everything. You're just showing one version, one type of memory.
Yeah, Ariel.
It would kind of hinge a bit on what we mean by decode, in the sense that if you're able to show that, like, you know, you have this slice, you found 1,000 different little modules, you show that you saw in this particular slice, you had your calcium imaging sequence, and then you saw the structural connectivity which lined up with that in the slice. You'd have found like the bits of data maybe that correspond to something interesting, in the same way that if you had a hard drive and you identified like the words and you could show that the sequence was regular, that like that was probably corresponding to data. But have you decoded it if you haven't pulled, like, if you can't interpret it, basically? And if it's just a slice with like a sequence, but you don't know what that sequence means, it seems like maybe you haven't actually decoded anything out of it, as opposed to like behavior, which is what we're using to ground our interpretation when we talk about memory decoding generally, of like, oh, that corresponded to a maze, a song, a tone association, something.
Yeah. I mean, I feel you're absolutely right, but I am willing to take the devil's advocate position at any time on this, because at the end of the day, I feel like there's the fundamental thing is that we have theories of memory, and we're not sure if those theories are correct. And so if I see something that nails it, just absolutely nails it, and it's like, oh my God, our theory of memory is really, really correct, but somehow it can't be connected all the way out. It's like, oh, this experiment A and experiment B together say everything that needs to be said. But somebody didn't do experiment A and experiment B inside the same animal. And so we have to wait for that. That seems more stubborn-minded of a criteria. But it's an ongoing debate on this. Any other questions? I know we're at the John.
Yeah, this is a bit, I don't know, orthogonal, but I'm not a neuroscientist here, so I'm going to ask those questions. So I know that Henry Molaison didn't have any CA3 when they did the autopsy later, so that got rid of one of the three theories for kind of what the hippocampus is doing, right? So now it's this so-called systems consolidation and multiple trace theory are kind of the two competing models for what CA3 is doing, if I got that right. And so my question is, what is this paper saying about
I mean
how this relates to long-term memory? What's the hippocampus doing? Is it
HM didn't have CA3, and that means that HM would not be able to make any new memories, because the first step in the consolidation process was cut off.
Yep.
And I think nobody's saying that well, maybe there are some people that are saying that there's long-term storage in the hippocampus, but I think to me, we're not really talking about long-term storage. But I think I see what you're saying. I mean, it's like well,
there's a secondary model called multiple trace theory, which says the hippocampus could still be involved in like traces to the long-term memories that have been coded in cortex, because Molaison had like not very vivid memories of the long term, and so maybe his traces were being like wiped out. My question though is that, like,
imagine the future.
What do you think's happening, or what do these folks think is happening after you've recalled this episodic memory, and like Randall was saying, you're playing through a series of them in your head? What's happening? What's happening to? Where does the compression happen? That's my question. Where does the compression to long-term happen? Must happen in cortex somehow? What do people think is going on?
You want to take that, Randall? I can take it.
John, by compression, do you mean the distributed storage?
Yeah, distributed storage into cortex, so that you don't need it when you put in the long-term memory. You no longer need the hippocampus.
Yeah,
right?
So there are definitely details there that I do not think have been worked out. But one aspect of it does have a pretty good, some good support, and that is the involvement of two things. One is the replay that happens during quiet moments and also during sleep. That the replay is something that enables reactivating areas in cortex that were involved with the episodic memories, and therefore allow them to form stronger connections. And then also, and this is important for the distributed storage, that potentially during REM sleep, but maybe also at other points, that you have what might be an interleaved reactivation of these patterns so that you have older memories and newer memories being reactivated in an interleaved way, so that you're not erasing old memory as you're putting new memory in a distributed manner, which is something that you can, lots of people have already shown this in artificial neural networks, that you need interleaved training if you want to do that, if you want to achieve distributed storage. There is something, there's something, by the way, about the previous point about whether or not hippocampus is still involved with longer term memories. We have to be very careful what we say or think we're saying when someone says these people had sharper memories or just vague memories, which is that I don't know those studies, so I can't comment on them directly. But I wonder if there's a case to be made that the sharper memory is really a case of updating, where you're having recall into CA3 and then you are forming memories, basically taking your old memories and creating new, fresh ones that maybe aren't even correct. You confabulate things. You're creating likely scenarios. That's something that we also discussed in our other journal club, obviously. Something that probably studies have to be very careful about, but maybe they were. I wonder if, there are some really careful studies of, say, maybe in animals, of how well they remember behavior precisely, if it's a long-term storage, if it's something that's, weeks later, months later, or not. I don't know. It'd be interesting to find those studies, yeah.
Thanks, guys.
Yeah. Samuel, go ahead.
Thanks. I want to go back to the previous question and then go on a slight tangent. So, linking an engram or a memory to a behavior, I am familiar a bit with the work of Susumu Tonegawa back when the headlines in 2014 were about implanting false memory into hippocampus. So the kind of scheme there was that they labeled recently active cells with CFOS, and channel rhodopsin for later optogenetic activation. But I'm wondering, I remember from that experimental setup, it took like 2 days when they took the mice off docks to have these cells express activity or be labeled with activity, which obviously will contain many, many memories in that time span. Does anybody know whether this procedure has improved over the years so that we can actually isolate something of the sorts of few hours of a neural activation?
I can't put my mind on it right now, but I know that there is a new technique that I wanted to do it for a journal club at some point that literally was trying to narrow that window down to it seems like Ariel knows what I'm talking about. Was it minutes?
Yeah, I remember we covered something where they did it with like some other light activation style system for tagging stuff that was only within like a minute or two of when they shone some light on. I'll try and find it. It was some other.
Instead of being doxycycline or whatever, had to have a light illuminating for the tagging to occur. And so that meant, boom, you could label it. I think it was developed at Janelia, but it's a good possibility.
So then my real question is, how does this contribute to an isolation of a behavior, provided that we can create a task for the mice or for any organism to do some memory recall? Is this a good strategy to try to get a discrete memory bounded in some way?
I haven't been able to think of a way of doing that. That's why I've been kind of more along the lines of auditory fear conditioning and stuff like that. So Ariel just said it's SC Flare 2. I don't have any thoughts on. I mean, it's a little weird. It's a great question because it's kind of like CA3 or hippocampus in general is a wonderful place to do these memory studies. And yet it's so high level that it's an association area, and so by its very nature, it's hard to trace back to sensory or to trace forward to motor in an easy way. So it may not.
What about quantitatively? Wouldn't it be a way to do something like you train two mice, one sensory deprived and one isn't? Could you say that you've got more sequential information stored, and then you could look at them
They've
after?
already done that though. I mean, if you're looking for like, is there more synapses or something like that? They've done those studies for a long time. So this comes back to the we want to decode a non-trivial memory, something that is non-trivial in the sense that it really pushes the boundaries of our theories and doesn't just doesn't give us a result that could potentially fit with a lot of different theories. Any other questions? I know we're way over time, so I'm always any other brief questions? All right. What's the next one up? Anybody want to say what the next one up is going to be?
I think I'm next, but I forgot momentarily what the paper was. Sorry about that. Don't have it handy right now. But anyway, this was great. Thank you for doing a paper that just solidifies the standard model. It's always good to do that from time to time. Yeah.
Okay. Thanks. Thanks.